Okay, so the plan for today is to spend much of the time discussing Hardy-Weinberg. Just to remind you, discussion sessions start this week. Some of you have had communications from your TAs, some have not. The graduate teaching assistants who are in charge of your discussion sessions. If you have any questions about what to bring to your meeting and you haven't heard from your TA, you should go ahead and email them for that information. Another logistical announcement, as some of you have noticed, and mastering biology is a gift because of the complications with the first 3 assignments, namely that we were on East Coast time, and so it slammed shut 3 hours earlier than anticipated here on the West Coast. I had mentioned before that I would eliminate the late penalty, but of course that doesn't do any good for somebody who's already used up their 3 attempts. So I went ahead and changed the grading policy. That's for the first 3 assignments only, just the 3 that were due last Tuesday, Thursday, and Saturday through Mastering Biology. There's no late penalty, there's no incorrect guess penalties, which means that these assignments are basically getting used to the interface and practice. If you don't actually do them, you will get a 0. So I do encourage you to at least do them, but you will not incur any penalties. But starting with the next assignment, we'll be back to the normal grading policy. If you're not sure what that is, you can click on the grading policy text as explained before. That should explain it. But anyway, also you may have noticed in the assignments there are a few new things that have appeared, namely the adaptive follow-up assignments. These are completely optional. What these are problems that are generated for you based on what you got right and what you got wrong. And so if you choose not to do any of them, that's just fine. There's no credit if you do them. There's no penalty if you don't. These are for you only. I'll be interested to hear from you. If you do find it helpful, again, the website is supposed to throw questions at you that are most closely related to those that you missed before, making it particularly helpful. And then just to let you know, the very last day to enroll in Mastering Biology is January 19th. After that time, the date stamp on enrollment, sort of that slams shut. Okay? Any questions about any of that? Yes, question in the front row. Thank you. Oh, the comment was, thank you. Yes, I get there was some sort of aggravation associated with the issues of the first time. You're welcome. So. Last time we were discussing the fascinating story of the coevolutionary story between what's going on with primates and also their lice ectoparasites. I'm just going to spend a few seconds finishing up on that interesting story, and I want to make a couple of final points. This phylogenetic tree that is in front of you right now is for the primates, not the lice, just the primates themselves. It looks like I have a pen, right? So again, just to focus, this is the primates here. You may notice something that's different from some of the other phylogenetic trees we're looking at, namely that there are times associated with specific events on this tree. So for example, the human-chimp divergence is thought to have taken place approximately 6 million years ago. So when you do see times explicitly on a phylogenetic tree like this, this does mean that there is some evidence via dated fossils or by molecular clock evidence that these are the approximate times of the divergences, and you can interpret it as being the best estimate for divergences. This is quite different from the kind of trees that we've been looking at before or diagramming that look like this. If there's no time involved in a phylogenetic tree representation like this, then you can assume that there's no implicit temporal component. So for example, you should not assume that the amount of time between this divergence point and this one, here I'll draw here, this divergence point and this one is not assumed to be approximately the same as the time between this and the present time. In a phylogenetic tree, such as the one in the upper left that I just quickly drew for you, if there is no time on there, then you should not take it quantitatively in terms of specific time stamps at all. The information in there is certainly showing the order, so clearly this lineage diverged from this lineage prior to this lineage diverging from that one, but you don't know anything specifically, quantitatively, about how much time, in fact, went by there. Go ahead and it's out of the way so it's not distracting. So now let's consider if we do have a case in which the two very closely associated groups have an entangled evolutionary history. Let's consider what the lineage for the lice would look like if, in fact, there was co-speciation, and not just between the humans and chimps, but, say, all of the different louse lineages. Actually, I'm going to switch to a different color that I think will maybe stand out better. Let's make the lice in bright green. So it hopefully will make intuitive sense to you that if I'm going to superimpose the phylogenetic tree of the lice, if there's a perfect match with the primate lineage, let's go ahead and just start down here. If when these two branches diverged, they took their lice with them, and they're now in completely different habitats and so on. And then again, we've got a divergence between these lineages. They take their lice with them and so on. One would actually expect that the louse lineage and the primate lineage would match up perfectly, just like this, if in fact every time the primate lineage diverged, the lice lineage diverged as well. In fact, you don't usually see representations like this one on top of the other because it just makes it really hard to see if one is on top of the other. I'm interested that you understand the interpretation of why it is that you would expect such a relationship. And of course, as mentioned before, humans actually have 3 different types of lice. And so if we were going to draw that on here, for example, perhaps it would look like this. It would look like this, so I've got 3 different branches then on the human lineage. It would look like this if divergence between those lines took place after the human louse lineage diverged from the chimp lineage. Is that in fact what happened? Well, there's actually a fair amount known about this, and so here is what the lineages look like. They're shown here in a more typical configuration where instead of being drawn right on top of each other, they're shown facing each other in mirror view. Notice that there's no time on this. This particular figure is actually from a scientific paper that I have supplied for you in optional reading. Should you wish to read up more on this, it is not required. I'm just supplying it as background information. So over here on the right, we have the primate lineage. We've got pictures of the different lineages. We've got a picture of Darwin here for the humans. And then over here on the left, we've got the louse lineages. And you can see that there is almost a one-to-one correspondence, except with one exception here, and that is the human pubic lice, actually, are not more closely related to the other human lice. So here I'll circle, this is the species for both the head lice and the body lice, the ones that live in the clothing and also associated with head hair. And that species is most closely related to the chimp species, as you can see with the dotted line connecting the chimp lice with the chimps and the human head and body lice with the humans. But we've got something funny going on here with the pubic lice, which are actually more closely related to the gorilla lice than they are to the other human lice. So what this suggests to scientists who study such things in terms of the evolutionary history of these ectoparasites is that at some point long before humans and chimps even diverged from each other, which is when the gorilla lice lineage had diverged from the other lineage, or subsequent to that, then we've had a transfer of the gorilla louse lineage back to the humans, because they're showing up here in the human lineage as this mismatch between the louse lineage and the primate lineage is shown here. So this transfer from the gorilla lineage to the ancestral human lineage then is thought to have taken place after the humans and the chimps separated out. This does not necessarily mean there was any kind of sexual encounters between early humans and gorillas. There are other ways of picking up these lice. So for example, louse transfers can occur because of predation. So if early humans were capturing and eating gorillas, for example, or spending time sleeping in their nests. That's another way of picking up lice and so on. But I bring that up as an interesting example of evolution in two groups. But before I leave this topic in general, I want to bring up one final point, which is molecular clocks. This is a topic that's not brought up in your book, but it is important for you to understand where the timing information comes from. This particular figure is actually from Kumar 2005. It's not from your textbook. The x-axis is time in millions of years, and the y-axis is nucleotide substitutions for a particular piece of DNA which is not identified in this particular graph. And this is a nice, straightforward, linear relationship between the y and x-axis, showing that as more time goes by, there are more nucleotide substitutions. And this makes intuitive sense at some levels, I think. And when for DNA stretches for which this relationship is true, it should make sense why this kind of information can be used to back calculate how long two lineages have in fact been separated from each other because of changes in the DNA sequence because of these nucleotide substitutions. If they're occurring at a more or less constant rate, then you can back calculate how long two lineages have diverged. And this is in fact one of the kinds of information, one of the sources of information used to try to backdate divergences. But there are problems with this, as shown in this particular graph, where though the x-axis is still time, now it's time since a divergence in millions of years, and the y-axis is percent of genetic divergence. And the main take-home message of this is that in looking at different kinds of molecules, we've got hemoglobin and cytochrome C and so on, they're not all lining up exactly the same. The rate at which there are substitutions is fairly dependent on what exactly that part of the DNA is coding for, if anything at all. And so if one is going to use a molecular clock estimate, then one has to first verify that the stretch of DNA that one is using does in fact have the relationship that one is assuming, and one can't jump from one to another. Okay, I'll go ahead and move on now. We'll consider the topic of evolutionary mechanisms. We've already learned about natural selection. That is just one of 4 different phenomena that are recognized as evolutionary mechanisms, meaning phenomena that can lead to changes in allele frequency with changes in generation. And these 4 evolutionary mechanisms are listed here: natural selection, genetic drift, gene flow, and mutation. I'll introduce them extremely briefly right now, and then we'll spend more time on them later. They all have different causes, and they also have different consequences. So natural selection that we've talked about a little bit already, this is when we have individuals with certain heritable traits that are more likely to have more offspring, either because they survive to reproduce, or when they do reproduce, they have more offspring. So they then are disproportionately represented in subsequent generations. And this leads then to adaptations, to fits between traits that have at least some genetic basis and the demands of the environment in which those organisms find themselves. So natural selection is the only one of these 4 mechanisms that actually produces adaptations. Genetic drift is a completely different phenomenon. This is something that students often find a bit confusing. This is a phenomenon that occurs primarily in small populations, and this is just the random fluctuations that can be driven by being in a small population. So these are just random changes in the allele frequency from one generation to the next, and we'll spend a little time talking about that later. Gene flow is just simply the movement of individuals from one population to another, and when individuals move, then they take their alleles with them. And so if the individuals that are moving are different in their average genetic makeup from either the population from where they're going, from where they're coming, where they're going and where they're coming from, then this can change or modify the allele frequencies in one of those 2 populations. So this can cause changes in allele frequencies, just the migration or immigration of individuals from one population to another. And then finally, mutation should be very familiar to you. This is the ultimate source of genetic variation, and this is what introduces new alleles in the first place. So we'll be going through all of these later, but first we're going to consider, spend some time formalizing how to think about allele frequency and allele frequency changes, which means we're going to consider Hardy-Weinberg. Hardy-Weinberg principle or Hardy-Weinberg equation is a way of describing a very simple case, the case when there is absolutely no evolutionary changes whatsoever. This principle or this equation allows one to make predictions about the relationship between genotype frequencies and allele frequencies of some gene that's under consideration in the absence of evolutionary change. And so if there's no evolutionary change whatsoever, then you know that the four mechanisms that we just discussed are negligible or are insignificantly important. So one can assume no mutation, no natural selection, no gene flow, and no genetic drift. If one is assuming that the Hardy-Weinberg assumptions are approximately valid in a particular case. Notice that we are just talking about a single gene, so this means that we can assume that these four evolutionary mechanisms are not occurring with respect to that particular gene under consideration. And of course, there's always mutation going on at some low level and so on. So the point is just that these four have just a very small magnitude so that one can in fact safely ignore them. And then finally, there's one other additional assumption that goes into use of the Hardy-Weinberg equation, and that is that within the population you have random mating, such that any egg is just as likely to meet any sperm in the population as a whole. That's a really important underlying part of the math. So let's spend a little time walking through this, but I find that one thing that helps in teaching this often is to go ahead and do a quick primer on calculating frequencies. And one of the important things to keep in note is it makes a difference if you're calculating frequencies. Are you talking about allele frequencies or genotype frequencies or phenotype frequencies? So we'll go ahead and practice calculating all of these one by one and in that order using a very simple small population for the basis of our calculations. And keep in mind that on all the tests and so on, you're not going to have calculators or computers or anything. I'll expect you to be able to do this by hand, so obviously the math will be fairly simple. We won't have really weird numbers or very large numbers, unless it's given to you as sort of a worked out equation or something. I'm very interested in identifying that you understand how to do it, not that you can necessarily go through complicated manipulations on a piece of paper. Okay, so calculating frequencies. Frequencies, that word has a number of different meanings. In the context of Hardy-Weinberg, specifically it means proportion or percentage of a total. And so if we consider a really simple population here, and we're just considering in the genetic makeup of these individuals, just one gene, and we have another simplifying assumption that there's only two alleles at this locus. And those two alleles are A1 and A2. Okay, very, very simple situation. We have 30 individuals total. So total is 30 individuals. 10 of our individuals are homozygotes at that locus for the A1 allele, so it's their A1, A1. This final column over here shows what the phenotype is, right? The characteristics that you can measure just looking at the individual. So we have 10 individuals that are blue in our A1, A1. We have 10 more individuals that are heterozygotes. They are also blue. And then we have a final 10 individuals that are homozygotes for the A2 allele, and they are red. Let's start by calculating the allele frequencies. So I think what often helps for a student is to start focus that you're calculating allele frequencies and not genotype or phenotype frequencies, is to first consider what are my alleles and make a list. That's pretty easy here. We only have two, A1 and A2. So for calculating the frequencies of A1 and A2, what do? We take the number of whatever it is we're considering and we divide by the total. So we want then to consider the number of A1 alleles and divide by the total number of alleles. And so how do we figure that out? The number of A1 alleles, well, we've got 10 individuals, each with 2, so that's 20 alleles from the A1 individuals, right? We've got 20. We've got a few more from the heterozygotes here. We've got 10 individuals, each of which has 1 A1 allele, so there's another 10 from the A1, A2 individuals, and of course the A2, A2 individuals have none, so we don't need to add anything else. How many total alleles do we have? Well, we have 30 individuals, each of which has 2 alleles, so we have 30 over 60, right, which is 0.5, right, or 50%. And then quick reminder, and don't be insulted I go through this, but I have to say a lot of errors on tests like this are people making mistakes going from the decimal to the percentage. Remember to move the decimal place over 2, right, going this way or that way, right? Okay, it's really easy to get that. Okay, so we can say the frequency is either 0.5 or 50%. So 50% of the alleles in this particular case are A1. To calculate A2 then, of course we can do this in one of 2 ways. If you notice that, well, they all have to add up to a total of 100%, then maybe the easiest way of calculating this is just to say 100% minus what we've already calculated for A1, which is 50%, right? The other way of doing this, of course, is to take the number of A2 alleles and divide by the total number of alleles. And of course, you'll get exactly the same thing, where you have 20 from the homozygote A2, A2s and 10 from the heterozygotes divided by 60 per usual, exactly the same thing. Okay, so that's allele frequencies. Let's now calculate genotype frequencies. Let's start by making our list of all the different genotypes. We only have 3, right? We have A1A1, we have A1A2, and we have A2A2. And so if we calculate the frequencies, this is the proportion of the total that are of this particular type. So we divide by the number, the number of A1A1 individuals divided by the total, right? So this is just simply what? 10 divided by 30. Our total number is 30 individuals. So this is 33% to the nearest percentage, right? 33 1/3. Same thing with our heterozygotes and same thing with our other homozygotes. So all of them are 33% in this simple case. Okay, last frequency calculation here is the phenotype calculation. We calculate the phenotype frequencies. Here's the phenotype right here. We only have 2. We have blue, we have red. To calculate the frequencies then, what do? We take the total number that are blue and divide by the total. So the frequency of blue and the frequency of red. Let's see, the number of blue individuals, we've got 20 divided by 30, right? So 67%. Frequency of red, 10 over 30 is 33%. Okay? So you should feel very comfortable with calculating for a given population. When you're given complete information like this, you should certainly be able to calculate allele frequencies, genotype frequencies, phenotype frequencies, and go back and forth between them when needed. Okay, if you're a little rusty on that, that's a really great thing to do in office hours with a tutor or in discussion sessions to get a little bit more practice on that. Okay, so now we're going to consider something slightly different. We'll go ahead and do this as a clicker question. Notice your answer counts, meaning you get more credit if you actually answer this correctly. This will be a background for Hardy-Weinberg calculations. We're going to consider one gene that has 2 alleles, A1 and A2. This is just a cross between 2 individuals, and I'm giving you the genotypes of the mother and the father. The mother is A1A1, the father is A1A2. When they have offspring, what is the best guess for the genetic makeup of the offspring? I've given you 5 different choices here. If you're not sure, great time to chat with your neighbor, figure it out, be friendly. Hello, my name is, I think it's this one, I think it's that one. I'll give you 45 seconds or so to work on this, and then I'll check back in with you. I'm hoping for 100% right on this. I would consider this pretty straightforward. Okay, 5 more seconds. Everybody clicked in? Okay, looks like it's stabilized. Maybe? All right, let's see what you guys think. Just hoping 100%. Well, close. Excellent, 94%. Fantastic. Okay, the 6% of you who missed this, you need to practice a little bit. One of the ways, of course, of doing this is to use something like a Punnett square. This is a Punnett square in front of you right now. Actually, in this particular problem, you were given that the mother is A1, so I'm going to fix it like that, right? And the father was a heterozygote as shown here. So what does this represent in a Punnett square? This represents the what you have in the egg and then the sperm, because if you assume the mother and the father are diploid and the gametes are haploid, then each egg only has one copy. But because the mother is A1 in this particular case, every single egg has A1 in it. So the only possibilities that one gets, so the egg can either be this A1 or that A1. In either case, then, if it combines with an A1 sperm from the father, you end up with A1, and then going ahead and filling this in. If it meets a A2 sperm from the dad, then you end up with a heterozygote as shown here. And most of you recognize that 50% of the possibilities are homozygous for A1, and the other 50% are heterozygotes. Good job. So in this case, 1 female, 1 male, and a simple cross, 1 gene, 2 alleles. We're now going to make the jump to considering a population as a whole. So we now don't just have a mother, we have all the mothers in the entire population, and similarly we have all the fathers in a population. And when one is considering this, then one considers all the gametes in sort of one large group without worrying about which exact mother or father it came from when we're considering just random mating, right? So if it's completely random mating, then any egg is just as likely to meet any sperm. You don't need to worry about which mother or father it came from. So when one is considering that particular case, then one usually uses the terminology of a gene pool to describe all the genes that are just grouped together. Now let's start with the same very simple case where we have just 1 gene and we have 2 alleles. And so those alleles are A1 and A2 at that particular gene. If we consider the frequencies in an algebraic notation, the algebraic notation for such manipulations are usually P and Q. And so P is the frequency of A1 and Q is the frequency of A2. And so that would be true both for the mothers and the fathers. It should make sense to you that if an A1 egg from a mother combined with an A1 sperm from a father, you end up with an A1 individual. There's 2 different ways of making a heterozygote. It could be an A1 from dad and an A2 from mom, or vice versa. And then there's just one way of making an A2 individual, and that is to get an A2 from both the egg and the sperm. It's actually fairly simple to calculate the numbers of such offspring in a population like this. You just simply multiply the frequencies. So for A1, then you just simply multiply the frequency of the egg, which is A1, time A1 in the egg, which is P, and then by the frequency of A1 in the sperm, which is also P. So you multiply P by P, and you get P squared. Similarly, in the upper right, you multiply P times Q. In the lower left, it's also P times Q. And then finally, in the far right, you get Q squared, Q times Q. So this is then how one calculates the offspring in the population as a whole, as opposed to the offspring just from a single cross of 1 mother and 1 father. So here now it's filled in for you, the Q squares and the PQs and so on. So let's now put some numbers on this. Let's consider that P is 70%, or in decimal format 0.7, and Q is 30%, or 0.3 in decimal format. So here you have 70% and 30%, otherwise it's exactly the same. So when we calculate p squared then, this is 0.7 squared, and this is 0.3 times 0.7, and 0.3 times 0.7, and this one is 0.3 squared. And notice, by the way, that of course p plus q in this case must equal 1 or 100%, because if it's not an A1, it has to be an A2, so you add up the frequencies and they must add up to 1. And of course that's true if you add up the frequencies for alleles, genotypes, or phenotypes. It always must add up to 100% if you're considering all possibilities in the situation. Okay? So the rather extraordinary usefulness of Hardy-Weinberg comes into the simplicity with which one can then relate the allele frequencies to the genotype frequencies if, in fact, all of the assumptions for Hardy-Weinberg are met. And that is that to calculate the genotype frequency of A1A1 is just p squared, or putting these example numbers in, multiply 0.7 by 0.7 is 0.49 or 49%. The heterozygotes, it's 2pq. Why 2pq? Because we've got one pq here and one there, and you add them together because there are two different ways of making a heterozygote. So the frequency of the A1A2 genotype then is 2pq. We're using these particular numbers. 2 times 0.7 is 1.4, right, times 0.3 is 0.42 or 42%. And by the way, just as p plus q equals 1, if you square each side and then expand, you get p squared plus 2pq plus q squared equals 1, right? Which are these three terms right here, right? Where the frequencies are p squared, 2pq, and then the last frequency for the final genotype, A2 is q squared, or in this particular case, putting in a numerical example, 0.3 times 0.3 is 0.09, or 9%, and check, they all add up to 100%. Yes, they do. We wouldn't be able to calculate or predict the genotype frequencies from the allele frequencies unless we could assume that any egg was just as likely to meet any sperm and that these allele frequencies were fixed. Okay, which is what happens if what? There are no evolutionary mechanisms, nothing to change the allele frequencies in the population, and random mating. So random mating would simply mean that any egg is just as likely to meet any sperm. If that is true, then there is this very simple relationship between allele frequency and genotype frequency. So this is then referred to as Hardy-Weinberg equilibrium. We're following the Hardy-Weinberg principle. So, if the Hardy-Weinberg assumptions are met then, no evolutionary mechanisms are operating in a non-negligible way. And then the relationship between allele frequencies and genotype frequencies are related as just described, p squared plus 2pq plus q squared. And furthermore, there are no changes in allele frequency with generation. And you can actually see that if you go back to this example here and think about, okay, we started with 70% A1s and 30% A2s, and then we had this totally random mating here. We can calculate the genotype frequencies. If we then calculate the next generation, we'll end up with exactly the same thing because we still have 70% A1s and 30% A2s for the next generation. So if we popped this into the next generation, this would get really boring really quickly because we just do the same thing over and over and over, getting out exactly the same numbers. Every single generation, we'd expect 49% A1A1s, 42% heterozygotes, 9% A2A2s. Put that back in, get the same number out. So as long as Hardy-Weinberg assumptions are met, then there's no changes in allele frequency with generation. It's the same generation after generation after generation. And there are no changes in genotype frequency with generation either. We start, in this particular case, we would expect 49% A1A1s in perpetuity if there's no evolutionary mechanisms and continuation of random mating. Notice that the population size can in fact change as long as it is always very large. such that, and the significance of that is just if the population starts to get very small, then genetic drift can in fact be an important evolutionary mechanism. But as long as the population size is above a minimum, then it can get larger. That is something that can change. You know, why is this such a big deal? Why should we care about this? Well, it provides a really powerful null hypothesis against which to compare what's going on in a population. You can look at the allele frequencies and the genotype frequencies and see if they actually do match with Hardy-Weinberg. If they do, then this suggests that there are no significant evolutionary mechanisms acting at least on that gene and those alleles in the population that you're studying. So this is a really powerful method that evolutionary scientists can use. So let's try out our Hardy-Weinberg manipulations and reasoning here. This is a more difficult problem than the one that we were just considering. It might take you slightly longer. Any response counts for this one. I would encourage you to work with your neighbors. Here's the problem. Consider a population of flowers, and 96% of these flowers are the color purple. What is the allele frequency of big A? And we've got 3 different genotypes here, and their phenotypes are described in this parenthetical aside here. So big A is purple, little a is white, big A, little a is purple. So I've changed nomenclature on you a little bit. So instead of calling the alleles A1 and A2, we're calling them capital A and lowercase a. And so this is 1 gene, 2 alleles, 2 different phenotypes. And I'm telling you that the population is, in fact, in Hardy-Weinberg equilibrium. So I'm asking you to calculate the allele frequency of big A then. So I'll go ahead and give you some time to chat with your neighbor and compare notes and see if you can figure out how to calculate this. I'll give you a minute. Guys all done? Too easy, too easy. Huh? Okay, well, it looks like most of you have sort of wrapped this up. If you're not quite finished, go ahead and click in your best guess here. I'll give you a few seconds here to click in. Okay, looks like everybody's clicked in. Okay. Let's see how many of you got this one. Oh, only half of you. Okay, well, as I said, it is a harder problem. There's a couple of different ways of doing this. Let's consider one way of doing it. Okay, what's the allele frequency of big A? One way of doing this is to make a list of the genotypes and the phenotypes like this. So here we have the genotypes. Oops, there's the genotypes. And then here's the corresponding phenotypes, right? So big A is purple, big A, little a is purple, and this one is in fact white, right? So we've got our genotypes and our phenotypes. And furthermore, we know that these two together are 96% of all of the flowers, and this one is 4%, right? So that's the phenotype frequency. So then how do we figure this out? Well, let's see. Let's say that P is the frequency of big A, and this is what we're actually trying to find, right? And then Q is the frequency of little a. And so we know that it's in Hardy-Weinberg equilibrium, so we know that the frequency of these different genotypes then would be p squared, 2pq, and q squared. So perhaps some of you did something along the lines of thinking of, well, then p squared plus 2pq must be equal to 96%. That's certainly true. That's actually a slightly more complicated way of getting there. You can get there eventually if you also combine that with p plus q equals 1 and so on. Now you have two equations, two unknowns, so on. But it's actually a little bit easier to just go ahead and start with this point here, recognizing that q squared is 0.04, right, is 4%. That's actually a little bit faster. If you then take the square root of both sides, then q is equal to 0.2. This is where sometimes people go astray. It's those darn decimal points, right? Or if it helps you, you can think of this as 4 over 100 and take the square root. Then this is the square root of 4 divided by the square root of 100, right? So that's 2 over 10. So in other words, it's not 0.02, right? So if q is 0.02, then how do we figure out p? Well, we recognize that q and p together must equal 1. So p equals 1 minus 0.2 or 0.8, which is the correct answer. Okay, so that's one way of doing that. So if you got that one wrong, then that would be a good one to maybe, a good type of question maybe to practice on a little bit, going back and forth. Okay, so as mentioned, the Hardy-Weinberg principle then allows us to make predictions about the relationship between genotype and allele frequencies in the absence of evolutionary change, so meaning no mutation, no natural selection, et cetera, et cetera. But it also includes an assumption about mating and random mating, and again, that should be obvious from the representation of the Punnett square-like format for describing the algebraic relationships. But let's go ahead and explicitly consider a case of non-random mating. And of course, there's many different ways one can have non-random mating. It could be that just one particular genotype is more successful at mating than any of the other ones, or you could have all sorts of different kinds of patterns. Let's consider a specific case in which all individuals in the population only mate with the same genotype. So for example, we would have A1A1 individuals only mating with other A1A1 individuals. A1A2 individuals would only mate with A1A2 individuals, and A2A2 individuals would only mate with A2A2 individuals. This would be an example of a non-random mating pattern. So we have basically 3 different types of mating configurations going on here. So why would this then result in deviations from Hardy-Weinberg? And down here we've got the familiar Hardy-Weinberg calculations shown before. Well, let's consider what this looks like. Here we've got A1 times A1A1, A1A2 times A1A2, and A2A2 times A2A2. So the 3 different mating types we can have if all individuals in the population are only mating with their own genotype. Then if we consider the case on the far left where it's all the A1 homozygotes are mating with each other, then of course 100% of the offspring are A1A1. Similarly, over on the far right, if we consider the homozygote matings for homozygotes at A2A2, 100% of all of the offspring will be A2A2. So far, it looks like you're just getting more of the same, right? All the individuals are just simply producing individuals that are genetically identical to themselves, which is true except for what's going on with the heterozygotes. So if we consider what's happening with them, we've got the heterozygotes mating with each other. We are ending up with 3 different distinct genetic types of the offspring. So we end up with 50% that actually genetically match their parents, right? We've got 50% heterozygotes, but we also have 25% A1A1s and 25% A2A2s. And so what this means is that there are fewer and fewer heterozygotes with every single generation because half of the heterozygote crosses are actually resulting in homozygote offspring. So if you follow this generation after generation after generation, you actually get this figure here from your textbook. This is generation 1, 2, 3, 4, and so on. You can see that in every single generation, then half of the proportion of the population that was heterozygotic at this locus is now gone in terms of representation of the population, and the total of the population that are the homozygotes is growing and growing and growing. Eventually, it should make sense to you that if you do this for enough generations, then you end up with 50% A1A1s and 50% A2A2s. And at this point, then, of course, it would no longer be changing, but you still don't have random mating, right? We would still have we would have lost all of our heterozygotes at this particular point in time. So if we go back then to considering Hardy-Weinberg and how this doesn't match up, it depends, of course, on what the allele frequencies are. In that particular case, we were looking at it was 50-50. These numbers are for 0.7, 0.3, but it's exactly the same reasoning and outcome. If one starts with 0.7 and 0.3 for the A1 and A2 allele frequencies respectively, then given enough time, one would end up with 70% A1A1s and 30% A2A2s. If they're only mating with the same genotype, there would be no heterozygotes. This is not what one would predict from Hardy-Weinberg. Why do we know this? Because we already went to the trouble to calculate the frequencies if there was Hardy-Weinberg. In true Hardy-Weinberg matings, then, one would expect 49% A1A1s, not 70%. So this is an example of how non-random mating by itself can cause the allele frequencies and the genotype frequencies to not be related to each other in a way that would be predicted assuming random mating. It causes a deviation. Okay. Yeah, actually, this is exactly where I ended at in my earlier section. We have 3 minutes left. I'm going to go ahead and let you go. I'll see you guys on Wednesday then. Have a good couple of days.